formulations, and then do optimization of various dependency of different products, and then development into specific products. So some of the products which are shared on the screen are right now, and the Nile and Chick are some of the brands, and we are a multinational company, and then providing products at least for 140 plus countries across the globe. And then these are some of the so I am looking after one specific kind of department which we have is a Radberry veterinary diagnostic, in which we are going to test different veterinary subjects, especially starting from dogs, cats, then parrots, for different diseases, as well as the DNA-based diagnostics. So I'm heading the division over there. So I'll take you through some of the work which we carried out during our interactions, and then taking up from some of the products which are required for developing a effective biological candidate for vaccines. Yeah, so why we need E. coli? Why is most favored organism for bioproduction? So cultivation of E. coli is very easy, and that you can take it up by plant and animal derived yeast extract and cas amino acids, amino acids. Then it was isolated in 1922 from diphtheria patient, and then it's in use for production of biopharmaceuticals, starting from insulin to various vaccine and then subunit vaccines. And to do production in E. coli is not a very difficult task. You can do easily by using codon bias and optimization, and then purification of the major proteins in either soluble state and insoluble state with a very low cost. And there are many new strains are getting developed and which can help you in properly folding up the protein and making of disulfide bonds. And they are really very well for proteins which are membrane proteins also, which are difficult to express in native condition. So what are the challenges we have? Why we want to do some kind of optimization to produce biopharmaceuticals over here? So it is specifically for a molecule which you want to develop, like it can be a metabolite which can be developed using synthetic biology by optimizing in metabolic pathway and all, and then proteins which we would like to do and develop for various diagnostics, vaccine, and therapeutic applications. So the challenge is each molecule is different, and it behaves differently, and then there's no set process you can use for doing it. And for different conditions, optimization, you need multiple number of experiments to be done. And although whichever conditions which you use for the previous product may not work for the new product or new molecule or new protein, doing single experiment at a time and then taking out the output out of that is going to be highly time consuming, and then is going to put a lot of effort over that. So what we need? So we need multifactorial design technology using proper statistical approaches, and then designing experimental processes, and based on the basis of we can do critical parameter optimization, and then do various dependency based on the experimentation, and then do a robust design which can give you freedom in terms of getting the product developed. And even after you develop further, you can get it done through various manufacturing processes also, because you will have defined tolerance as well. So what do you want to do? And so that typically in experimental design process, you need to have a statistical approach, conformity to the regulation, like linear model or model where you can see the dependency of one process on the other, and then reduce the number of experiments to get a good outcome by using different level of factorial designs up to from two factors to multiple factors. So why when we do work on E. coli, to scale up, we need to do a fermentation, and then fermentation is being carried out for different molecule based on the requirement you have, from 2 liters to 50 liters in a pilot scale. In industrial scale, it is again going to be at a higher scale. But when typically OD is 10 to 40, then and based on the product yield which you are going to have and conformation and stability, the process is batch fermentation, which is going to complete in 8 to 12 hours. So these are the parameters which you need to optimize for that. One is the in terms of media, and what is the temperature for growing the culture, and then what is the time for induction, and ultimately what is the time for protein production. And same with the batch, when until 8 to 12 hours, when batch is completed, it is taken as a fat batch mode, in which you are exhausting the carbon source. And when it is indicated by the increase in the dissolved oxygen level, which is reduced, and then your DO is going up very high. And so that you are going to set up a specific growth rates, start ranging from typically doubling point time of 0.5 to 0.12, 0.22 mu per hour. And here again, you need some optimization using glucose, buffer, and trace elements. And then what are the things you need to make sure that our constraints for the process are acetate, format, and succinate. So there are three processes which are involved. One is the screening. First, screening, you need to screen different sources, and depends on the type of process which you are doing. If you are doing a process for a pharmaceutical development, then you need to look for a optimized sources. Likewise, you need to have a process defined material to be used, like in standard companies. And when you're doing a biofertilizers or using some enzymes production, then again, you can go for a cheaper sources of carbons, and then complex nitrogen sources. It can be either sugarcane molasses and some digest of different proteins, which are coming from as a waste product from different places, where it is going to be metabolized into develop a full process. So first of all, you need to identify what are the sources you have, and then screening which are the type of fermentation you are going to do, batch, fed batch, and then time required, and what is the process we are going to use. So it can be semi-defined and the complex medium, and then screening of the sources. Then simultaneously you need to do characterization of the various processes by defining different amounts of nutrients which you have, and then what is the time for fermentation, and then when you're going to go for fermentation, you need to find out whether you're getting outcome as a soluble or insoluble protein. Mostly the people do prefer getting a soluble protein, because refolding is a difficult process and it requires larger volumes to be processed. And once that is optimized, then you go ahead and do concentration optimization of various processes using a multifactorial design with the proper dummy experiments. And that can be done not at fermentation level, but you can do it at shake flask level also. The typical outcome is going to be measured by either optical density or the product. A product can be evaluated by directly lysing the cells and then running it on a FPLC or HPLC into column, and optimize the concentration which is going to be obtained from that process. Yeah, if then optimization is complete, then you go for some 2-3 batches, and then you can take it up in a shake flask, and then final optimized is done by proper inducer concentration using DOE processes in the fermentation process. Yeah, so there are various conditions which you need to. So first step is screening. Then you need to find what are the known factors and unknown factors. Second is the characterizations. What are the factors which are affecting, and then what are the interactions you may have. And then different optimization processes are being applied, which are response surface methodologies, by placket vermin, and reforms for surface methodology by other methods and artificial neural network, genetic algorithm, needle. This depends on the type of process and type of parameters you have. You are going to do it further. So are we going to do a typically high-end statistical approach for doing this? Now, there are certain softwares and then things which are available right now, which can be applied and doing it. So DOE and states are the licensed software. JMP and Minitab are also some licensed software, but we can also develop a Python kind of a notebook, which you can use for utilization for different kind of responses calculation in the statistical platform. So we did some of the optimizations for different proteins, which are specifically for malaria vaccine candidates, in which we initially were getting about 24 mg per liter yield, and then after optimization of different conditions using different cell types which has disulfide bond isomerase, we were able to increase the percentage of the protein production by 270%. Similarly, we did for multiple proteins and all, and this we did optimize inducer concentration, time post-induction, and product yield. So except for last two proteins, all proteins were obtained as a soluble proteins. So what we need to do is that, we designed a block of different factors which we are going to use, and then we did a shake flask experiment in which we did 25 conditions together and find out the response of all those together. And then the response which we calculated for mixing together yeast extract, select sorbitol, and glucose in a ratio which is being optimized and defined by our factorial approach. And we had 3 blocks for that, and we recorded the response in terms of optical density after the completion of the process. And then we did look for the response on the product yield, that is going to be mg of the protein in per liter in the shake flask. So this is what is the correlation we got it for between the product yield and optical density. So if we are going to have higher optical density, we are going to get a better product yield. And again, we did estimated and then checked out whether is what are the parameters which are going to fit in our standardized plot and all. So in a half normal probability, we are going to get most of the events which are coming up are going to be positive effects, which are not coming up under half normal probability plot. So if we consider which are the positive effects, the t-value, they should be above the t-value limit of the responses. So we are able to find out the positive effects, especially with the yeast extract, were above the t-value. Again, we were able to look for what are the various t-normal plots for the residuals, and then we were able to find out that most of the responses and the probability for the residuals were coming in the perfectly normal statistical configuration. So we did studied for the residual and versus the 3 different blocks. So we were able to find out again the values which we were having in the 3 replicates were almost coming out to be similar. And then further, we did the Cox plot and then find out whether our values for the model are coming up into the right configuration and right conditions or not. We were able to get the probability in between of the residues between minus 3.5 and plus 3.5. Again, the Cook's distance is a major where you would say that whether your experiments which you are performing are normal or which are not, or we are having some extra or exploratory or maybe some outliers in there. So we were well below the outliers and then found all the relevant experiments in the right place. So this is one of the example of doing it. So we were able to do the constants also. And so we did look for the correlation between the different substrate which is going to be used for fermentation. And similarly, we were able to select different conditions. So the best or an optimal condition, which is going to give you the least pricing as well as the better result, is selected in the number one. And all other conditions, if we are going to apply, we are going to get a similar product. So we were able to select based on the statistical evaluation, and then we were able to find out how we are going to move ahead. Similarly, so I was only able to showcase you about the media optimization. So there are other procedures which are going to be taken. The slides look too many, but when we are actually doing the experimentation at that time, that is the only time which is going to be taken up by the processes. Other than that, you can easily carry it out by using different softwares and maybe using the Python algorithm. So future strategies now for different uses of codon and eukaryotes and eukaryotes, do affect the expression of the heterologous proteins. So in that case, there are some amino acids which are not being produced or which are not having codons in the E. coli, which are easily going to be supplemented in the medium and which can be taken up easily for production and increasing the protein. And then rare codons which affects the protein production and mRNA even plasmid stability are again going to be taken up care by design of experiments approach. And then using the tRNA pool which are not present in E. coli, and then which can be utilized to increase the growth of or increase the product for the different fermentation processes. And there is also optimization can be taken up, likewise considering a batch or fat batch or different media components to do and find out which is the actually cheapest processes which can be optimized. And again, we can do further use of carbon, nitrogen, and other conditions which are required for batch and fat batch conditions to reduce further. There are several algorithms which are being studied in the placket Vernon, Volkswagen, Taguchi design, central composite design. And these are all depend on number of experiments people would like to do. So there are different algorithms which are applicable. If you are going to multiple factorial, you will go with placket Vernon. If you are going 13 to 15 experiment, then Volkswagen. And again, the Taguchi and central composite design again depends on the type of response you are going to get. So most of the softwares do have it. And other than that, you can do response surface methodology and ANN using the Python algorithm, which are easily available, and then they are open source. Yeah, so this is one of the publication which we had regarding all these algorithm and then processes which are going to be applied in detail. You can refer to that. And I'm open. Thanks for providing opportunity for me to come and present some of our experimental design and processes in the conference, and open for the discussion for the questions if anyone has.
Thank you so much for the wonderful presentation there. Congratulations for the publications as well. It was very nicely presented. If anyone has any questions for Dr. Puneet Gupta, please go ahead.
So I'm helping people who want to do Western industry related processes development and all. And so some of the activity which we are carrying out and some of the things which are free also for new learners and students also. So they can contact us and then we can do a full session and training on various approaches to be taken up for getting the highest product yield and then improving the process which they already have. So,
thank you so much for sharing that with us. I'm sure that would be very useful for people who want to pursue that. Thank you for your presentation. Thank you for sharing your data with us and your wonderful presentation with us.
Thank you. Thanks for giving the opportunity for presenting. Again, I would like to thank the organizers as well as the committee. I was supposed to be in conference physically, but maybe due to some time constraints, I was unable to come and join. Thank you very much.
Thank you. Our next speaker is Sayani Bhattacharya, Krupa Nidhi College of Pharmacy, India. The title of the